Thanks, Tanya. Good morning, ladies and gentlemen. Thank you so much for joining us today. My name is Jennifer Stone. I'm the Market Manager for Cancer Genomics at Illumina. And today it's my pleasure to share with you a presentation entitled Illumina's Genomic Technologies, Applications in Cancer Research and Translation. The goal of this presentation is really to communicate Illumina's view on cancer research, how we're pushing the technology forward to make the very best tools and put them in the hands of the research community so that discoveries can be made as quickly as possible. So I'll review Illumina's approach to cancer, some of the things that we're working on from a research perspective, and then share a few anecdotes of how our collaborators and our customers are leveraging these technologies to make discoveries that eventually, and in some cases, maybe sooner than we all think, move into the translational space and start affecting and positively impacting patient care. So the agenda for my talk is pretty simple. We're going to talk a little bit about a lifetime of cancer, and this is a framework that Illumina is using to really understand the myriad different research projects that are going on in the cancer space. We'll then talk about what we're doing about it and where we are today. And those two questions really tee us up to talk about, again, the research that Illumina is investing in, how we're pushing our technologies forward, and then also provide some anecdotal stories of how our collaborators and customers are making real-life discoveries today. So lifetime of cancer, this is simply a framework that we're using to simplify the different bins of research activities that are going on in the cancer space today. This isn't a framework that we invented. Many thought leaders have presented on something similar to this, a concept that allows us to understand the different points along a hypothetical individual's life where genomic information eventually has a potential of impacting positively their medical experience. And so we're all born with some level of susceptibility for cancer, germline risk. We then hopefully proceed through our early years healthy, but go in for regular screening for various things from cholesterol to just making sure we have healthy height and weight. Then in this hypothetical individual's lifetime, perhaps there's an event of cancer. 1 out of 2 men, 1 out of 3 women are going to experience this. And at that instance, there's an experience of diagnosis, profiling, staging, etc. Treatment decisions are being made. And then hopefully those treatment decisions are successful and that person goes into remission or is cured. But there's always that specter of recurrence or a new cancer event coming into that person's life. So at each one of these stages, there's an opportunity for genomics to play an important role in providing information for understanding that person's risk, understanding their healthy status, understanding their cancer better, and then monitoring for the recurrence or new presence of disease. Now, in the research space, there's been a huge amount of work done in understanding germline risk. Probably the most famous gene that we all know and love is BRCA1 and BRCA2. But as we proceed through the years, coming into the GWAS era, most recently, scores of loci have been identified that contribute to an individual's population-level risk for particular cancers. Most recently here I have the headlines from the COGS consortium that published a suite of papers last week, leveraging a custom array that they designed in over 200,000 individuals. But in this space of the research of cancer research, really numbers are the game. Statistical significance is the game. And for that, we need robust, cost-effective technologies to really sample a large number of patients and draw those statistical conclusions. I'll jump forward a bit to the profiling, diagnosis, and treatment stage of this lifetime of cancer. And this is really an area where there's been a huge body of work done. Most recently, the information coming out of the TCGA group, for example, and I have some of the papers that they've recently published up here, where the goal is to profile, okay, what does the cancer state look like? What are the variants that are present in that particular tumor? What does the expression profile look like? Really understand the picture of disease for that particular tumor type. And from that, be able to draw hypotheses about how one can approach that tumor to really combat it, to combat the clonal evolution of that tumor, the diversity within that tumor, et cetera. All of these are very exciting opportunities for genetics and genomics to play an informative role going forward. Stepping to the next stage of a lifetime of cancer, response, recurrence, minimal residual disease. There's fewer papers in this space of leveraging the high-throughput genomic technologies to inform this particular stage, but there's some exciting stuff that's come out recently. The top paper listed here is the analysis of circulating tumor DNA to monitor breast cancer metastasis, which I'm sure you all have seen. But very exciting proof of principle study that illustrates the potential here. And then jumping back to healthy screening, which I think is an area of promise, but one where the least amount of work has been done. So in this instance, I'm actually calling out a specific paper that came out recently from Dr. Diaz's group, we're fortunate enough to have with us today, who's going to expand upon this work for you. So that's a lifetime of cancer. And so what is Illumina? What do we do in this space? Well, we have three pillars to what we're trying to do here. The first one is to continue on our tradition of supporting research with the very best, highest quality technologies out there to drive a critical mass of information that allows for ready interpretation and utility of the data. The next pillar, and this is a pillar that I think that is really driven home by our translational and consumer genomics business unit at Illumina, but this is to partner with leaders in the field to bring this genomic information as quickly as possible and as responsibly as possible towards the clinic. And then lastly, the last pillar is to work in partnership with the research community, with the medical community, to seed an environment where genomic technologies are going to be allowed to fulfill their promise in terms of bringing that information to bear on patient care. So the genome is, well, there's a lot of popular press about this, there's a lot of people that speculate this, but it's not a foregone conclusion as this cover from Time magazine would suggest. Everything that we are is not wrapped up in our DNA. And especially is the case with cancer, where it changes over time, it evolves over time, and we're going to hear more about that later on in the talks today. Now, we've already discussed in a hypothetical person's lifetime all the different points in time when genomic information could benefit the understanding of their particular healthy or diseased state and inform their treatment. These are all things in the future, and I think we can see the writing on the wall. But in order to get there, there's significant experimental challenges that we all have to face in order to achieve that potential. The first one is there's simply a wide spectrum of mutations that we need to be able to reliably detect and then catalog, and then ultimately understand their importance in the context of an individual. And then there's simply difficulties of detection. So cancer samples are by nature very challenging to work with. FFPE, for example, degrades the DNA, but we need solutions to overcome that so that we can use those samples and learn from them. Clonal heterogeneity, tumor evolution, all these things make cancer very much a moving target. And so repeated and deep sampling of a number of different individuals with cancer, as well as different, within an individual, different samplings of that person's normal and cancerous state will be important. So with that as a long-winded introduction, I'd like to jump to what we're doing about it and some of the research projects that we have ongoing to address some of these technical challenges that have to be overcome. So in addition to just thinking about cancer from a biological perspective, we're actively pushing on 4 areas that practically impact all of your research. It's time, money, and quality are the first 3 that I'll talk about. But these are very practical concerns. These are concerns of every single researcher, every single research program out there, the funding bodies, et cetera. And what we want to do is deliver as much as we possibly can in each of these areas to give you the tools that you need to make these discoveries. So starting with time. Our goal here is to continually strive to improve. This is a moving target, and we certainly want to maintain the best tools out there and push hard on them so that we can all achieve this promise. So in 2011, just for comparison's sake, it would take about 8 weeks to do a cancer genome. So sample prep takes about a day, the sequencing itself takes about 10 days, you throw on a couple weeks for analysis, et cetera, et cetera. It's a long process. In 2013, we're targeting a much shorter turnaround time for that. So advances that we've made in sample prep really shorten that investment up front to a few hours, down from a day. Getting the sequence data itself with the HiSeq 2500 can be done in just over 24 hours. The interpretation and analysis and informatics side of things is something that we are pushing very hard on, and we've been able to shrink that dramatically to a little over 2 working days as opposed to multiple weeks. The interpretation, we've partnered with leaders in the field here, for example, Gene Insight here, to help with the annotation of variants that people identify. And then ultimately this leads to a custom report. Our goal is to have this down to 48 hours. We're not there yet, but we want to have this process with very, very rapid turnaround time so that you can get to the information that matters as quickly as possible. It's not just turnaround time, but it's flexibility as well. And so I've called out a couple of examples of how we're trying to increase both the turnaround time, decrease the turnaround time and increase the flexibility of our solutions out there. And the HiSeq 2500, I think, is a great example of this. With the 2 modes, the high output mode, you can do many larger projects, fewer runs, but get a ton of data in one go. It also has the option to do a shorter run. This is the rapid run. So you can do smaller projects, get results quicker. The run time is a lot shorter. And so you have the flexibility to change up what project you're running on a sequencer much more rapidly. So you can pick and choose depending on what your research calls for at that particular time. The other way we're pushing hard on turnaround time and flexibility, for example, is with the Nextera Rapid Capture Exome that was launched a few weeks ago. On a HiSeq, this can go into the 2 modes, the high output and the rapid run mode, where you can do a large number of exomes in an 8-day turnaround time, for example, or a smaller number of exomes in just under a day. So depending on what you're looking at. It's also available on the MiSeq. So if you only have 1 exome and that's really what you want to look at very quickly, that's a possibility as well. So choice, flexibility, and turnaround time, pushing hard on developing that. So with Nextera Rapid Capture, you can see what used to take many weeks as a research project, you can now get the data and start analyzing it in a much, much shorter time. So moving on to the second aspect where we're pushing hard, this is on price. I'm not going to dwell on this because everybody's seen this graph. This is taken from the NHGRI, the dramatic reduction in cost over time of sequencing data. And these correlate well with some of the technology developments that Illumina has put on the market over the past couple of years. Suffice it to say, we're continuing to push in this area as well. And then the third pillar is quality. And so even for our own sample prep, TruSeq, which has been a standard in the industry for a few years now, it's not perfect. And up here I have a read alignment graph where it required 1 microgram of input DNA, PCR, gel, laborious protocol, and plus there's gaps in this particular region of the genome, high GC content. This year we launched TruSeq PCR Free, which addresses many of these pain points in a research protocol. So much lower input, no PCR, no gel, and as you can see, we've addressed the GC bias here, and there's a very reduced number of gaps across the genome. So our goal here is to come out with the tools needed to create a gold standard genome. This is just a detail of that. These are different classes of genomic features. Some of these are easy to sequence, some are not. And the ones in the red box are the classes of evil that we call. And as we go from the purple through to the blue bars, going from left to right, these are different versions of our technology. So even addressing our own products on the market, we know we can improve, and we are investing to make sure that eventually, as I said, we have the gold standard genome, the very best quality out there. We also want to make it easy. So in addition to these 3 pillars, we want what is right now a very specialized area of expertise, a specialized research approach, which is genomics. We want to make it accessible for people that aren't necessarily bioinformaticists, people that are really encouraged by the promise of genomics, but perhaps need a much more accessible way to go after this type of information. So one of the ways we're pushing on ease of use is to implement sample-to-answer workflows. So there's many, many steps going from the raw sample all the way to an answer or some sort of result that you can take into a follow-up experiment or take into some downstream application. We want to improve all aspects of this. And so the first one is accessibility, especially for cancer samples. FFPE samples, as I've already mentioned, but also very, very small input amounts, fine needle aspirates, et cetera. We are developing solutions that will unlock those samples for researchers to access. And then, of course, speed and efficiency, which we've already discussed, quality as well. But genome mobility, and by this we're referring to the actual data. So providing solutions such that it doesn't take forever to load a genome onto a hard drive and then you have to FedEx it to your collaborator. Developing cloud-based solutions that can potentially address those concerns. Ease of analysis, and we'll talk about that in a second, and interpretation. All of these are areas where we hope to make things as easy as possible. And this is a moving target. It's not going to be solved overnight, and it's going to evolve over time, but we're investing there. And then this last picture here is just to refer to reporting. So how do you take this information collected in a research environment, aggregate it, and then eventually take it to the next step of translation? Ease of use, I alluded to it, solving the informatics bottleneck. There's a huge amount of information out there. There's public resources, there's databases. How does one query all of those in an efficient way to come up with an annotated genome that can be used for downstream purposes? We're working to build the solutions that aggregate that information, leveraging cloud-based computing to make it accessible to everybody, even if you don't have an IT department. And then ultimately, again, come out with a report that can be used in any number of different settings. So that's just a quick insight into some of the areas where we're investing, where we're pushing hard to improve our technology, again, to deliver the tools into your hands to make these discoveries. So where do we stand today? Well, we're building the reference. We are definitely participating in a number of different large international projects that have been going on or are just starting up to really amass the total amount of information that we're all going to need to accurately and robustly interpret genomic variation on an individual level. So getting the sequence, well, that's been done. Understanding the protein coding function, non-coding function, all the different categories of variation, understanding those that have clinical relevance, pulling all this together is something that we are working on. We're trying to provide the tools to the community, including those very large projects that are developing these databases. The other thing that's very clear in terms of assessing where we stand today is that the non-coding information is extremely important. There's been a number of projects that have focused on exomes out there. That's a great start. It's very good for focusing on those variants that most likely are going to have a functional impact. But the latest papers to come out are really suggesting that we need to push deeper into the genome. We need to expand that view and really understand the variants that are having potentially more subtle effects on gene expression or a risk, for example, across the genome. And this is a paper that has highlighted the TERT promoter genes with an incidence that is equivalent or approximately equivalent to the BRAF mutation. So it is an important player in melanoma. The other thing, sort of the state of the state, is we need to go beyond DNA. So there's a huge amount of research that's been focused on DNA, but we need to bring RNA and epigenetics to the table as well. So RNA is important. I think that goes without saying, perhaps, but it's a great way to confirm genetic mutations that you're finding in the DNA, to find fusion genes. It's an excellent approach to do that. Find expression changes, which many people have been doing for years with microarrays, but doing it with sequencing is a very clean and robust way to do that with great results. And of course, the caveat, though, is that the interpretation of this requires normal tissue, adjacent normal tissue, which can be somewhat of a technical challenge. The graph here is a nice integrated study that showed the analysis of 17 lung cancer samples whole genome sequencing and RNA sequencing information. Of course, they did the tumor and the adjacent normal tissue comparison. And they found that 40% of the genomic mutations were actually expressed. So these are the mutations that were found in the DNA sample, but not in the RNA sample. And so if it's a variant that's in the DNA, but it's not in an expressed protein, that brings into question the relevance of that particular variant. And then for this particular study, they found a number of different druggable targets. But the graph here really shows that on the tumor allele frequency, so these are variants that are being identified from the DNA, are not necessarily variants that are being identified in the RNA. And so this helps researchers prioritize which variants to follow up on. So the next example I want to show is for glioblastoma, and this is referencing some of Charlie Swanton's work that was done in collaboration with our Chesterford office in the UK. But this is really showing that in this particular glioblastoma patient, not responding to treatment, they sequenced the genome, and within 10 days, they were able to have information that was important for this particular individual. They sequenced different biopsies and saw the differences in the tumor, the heterogeneity within the tumor. And this had important implications for understanding which drugs this particular individual would be most responsible for. So at least in Europe, there's very clear translational aspects of the work that is going on here. And then another example from Europe, this one is for CLL. Chronic disease, many treatment cycles, many relapses. I mean, this particular study by Anna Hsu, the whole genome sequencing was done at each major stage of this particular patient's experience with this disease. At each of these time points, A, B, C, D, and E here, we were able to catalog the different classes, the different clones within that particular tumor sample. And so you can see response to treatment, but even in the remission state, when we blow that up, with deep resequencing, we're able to see that there are, in fact, still tumor cells present. And in this case, we would have the profile of then what became the treatment-resistant clone, the purple one here, that led to recurrence in this individual. And so monitoring the course of treatment, the presence of the different clonality within an individual over time through their course of treatment could potentially identify remission clones that are going to cause a recurrence event earlier in that particular patient's experience. And so this is very exciting, very promising work. And then the last anecdote I want to share with you is using cell-free DNA as a non-invasive way to monitor treatment response in an individual patient. And this, again, is work that Charlie Swanton has worked on with our UK team. But basically, this is a lung cancer patient shown to have an EGFR gene mutation. It was treated per standards. The plasma from a simple blood draw does include tumor DNA. Great source for trying to track this because it's non-invasive. And deep amplicon sequencing of this particular patient's DNA allowed for the monitoring both pre and post of the treatment for the presence of the disease-causing mutation. And so here we have in the normal sample, we've got about 88% of the wild type. And in the tumor sample, about 12% post-treatment, the tumor mutation drops down to about 0.4%. So we can really monitor the success of this particular patient's treatment regimen. So in summary, I think we certainly believe, and I'm guessing everybody who's attending our session also believes, there's huge potential for genomic information to have a positive impact at many points throughout a hypothetical patient's lifetime. But there are still significant technical and interpretive hurdles that remain. We are trying to do our part by pushing hard in collaboration with the research community on the development of tools that will be needed to overcome those hurdles, to make the very best tools, the most flexible and accessible tools out there so that discoveries can be made. And we are partnering with collaborators to develop the critical mass of information that's going to be needed for appropriate interpretation of genomic information on an individual level. And I hope through some of the vignettes I shared with you, it's very clear that there's a number of research groups out there that are doing great proof of principle studies right now that are clearly showing and demonstrating the promise of genomic information, certainly in a translational, with an eye to the translational application of that information. I do want to highlight again that there's a number of us here today. We will also be here throughout AACR, so there's a number of opportunities. If you do have any questions or you have some research ideas that you want to bounce off of us, we're always looking for great collaborations. Please approach us. Let us know what questions you have. We're happy to help. We're happy to support you in your research endeavors. So thank you very much for your attention, and thank you for joining us today.
Does anybody have any questions for Jen? Any questions? We've got 2 microphones up there, and I also can run this around too, so she can hear your question. All right, let me start then. In one of the very first slides, you had that cancer continuum, and there was one region I thought you said had like fewer papers in response, and that's the response to reoccurrence and minimal residual disease. And do you think it's because of the technical or interpretive hurdles, or is there a reason why that particular area didn't have that flood of research that the other different areas had?
That's a great question. I think my gut feeling on it is there are some more significant technical hurdles in that area to do it robustly, non-invasively, and in an effective, efficient way. But I think we're pushing on that, and clearly folks in this room are pushing hard on that as well, so it's just a matter of time.
Jen?
All right, well, thanks everybody.